A5 and A6, AAQ Medical Science Unit 1. So this is cell ultrastructure and cell transport. So cell ultrastructure, recognise cell structures from photomicrographs or electron micrographs, calculate magnification and true size of organelles or cells from photo and electron micrographs, comparison of use of electron and light microscopes limited to consideration of magnification, resolution, ease of use and ability to observe live living specimens. Transport of substances into and out of cells. You've got active transport, use of membrane proteins and ATP, passive transport, simple diffusion, facilitated diffusion, osmosis, bulk transport, transport of substances too large to pass through the membrane, use of vesicles and ATP in exocytosis and endocytosis. So the magnification equation is I am, I at the top, am at the bottom in the triangle. I is image size, M is magnification, A is actual size. So the equation to calculate magnification equals image size over actual size. How to turn millimetres into micrometres. If you get given it in millimetres, you have to times it by 1,000 to get to micrometres. So if you're looking at this image, the actual thickness of the leaf below is 2,000, but the image is 50 millimetres. So you'd have to convert that into 50,000 micrometres, and then you do image over actual. So image would be 50,000, actual would be 2,000, so 50,000 divided by 2,000. And in the magnification equation, image size is the thing that you actually measure with a ruler in millimetres. So you measure it in millimetres, and that's your image size, your eye, but you have to times it by 1,000 to get it to micrometres. So microscopes, light microscopes, and how they work. Light passes up through the specimen and is focused by the objective and eyepiece lens. Sometimes staining is required. Advantages of a light microscope: they are cheap and portable, and you can view living specimens. Disadvantages of light microscopes is a lower resolution than an electron microscope. Resolution is limited by the wavelengths of visible light. So lower maximum magnification of times 1,500. How transmission electron microscopes work. Electromagnets focus a beam of electrons through the specimen. Denser parts absorb more electrons, so they appear darker. How scanning electron microscopes work. A beam of electrons scans over the surface of the specimen. Reflected electrons form an image. 3D images produced on the computer screen. Advantages of electron microscopes: there's a higher maximum magnification, approximately times 500,000. Higher maximum resolution, approximately 1 to 5 nanometers. Disadvantages of an electron microscope: specimen material must be dead as it is placed in a vacuum, and the image is produced in black and white, although it can be artificially coloured. So transmission electron micrograph on an animal cell, you've got the nucleus, which contains a DNA, nuclear membrane, rough endoplasmic reticulum, membrane, mitochondrion, Golgi apparatus. Transmission electron micrograph from mitochondria. You can see the cristae in the matrix. Scanning electron micrograph of blood cells. Remember, scanning electron micrographs are in 3D because they scan the surface, whereas transmission electron microscopes are a cross section, so they look 2D. Resolution definition: the ability to see individual objects as separate entities, so the ability to distinguish between 2 points. Magnification definition: how much bigger the image is than the specimen. Now looking at organelles, so we've got the nucleolus in the middle, chromatin, nuclear pore, nuclear envelope, double nuclear envelope, and all of that makes up the nucleus. You've got centrosome with 2 centrioles close to the nucleus at right angles to each other. Lysosome, mitochondrion, rough endoplasmic reticulum, microvilli if it's an epithelial cell, Golgi vesicle, Golgi body or Golgi apparatus, microtubules radiating from the centrosome, ribosomes, cell surface membrane, cytoplasm, and smooth endoplasmic reticulum. So eukaryotic cells contain a nucleus and other organelles that are bound by membranes. The plasma membrane is a selectively permeable phospholipid bilayer forming the boundary of the cells. Cytoplasm, a jelly-like fluid inside the cell in which the organelles are suspended. Chemical reactions occur there. Nucleus is surrounded by a nuclear envelope that has nuclear pores to allow movement of molecules through it. The nucleus contains chromatin. The function is to regulate cellular activity. The nucleolus is found inside the nucleus and produces ribosomes. Rough endoplasmic reticulum, the region of the endoplasmic reticulum that is studied with ribosomes and engages in protein modification. Smooth endoplasmic reticulum is an endomembrane system where lipids are synthesized and toxic substances are broken down. Golgi apparatus, a system of membranes that modifies and packages protein. Vesicles are small membrane-bound sacs that move products into and out of the cell. Lysosome is an organelle containing digestive enzymes. ATS ribosomes are larger ribosomes found in eukaryotes and the site of protein synthesis. Mitochondria is the site of aerobic respiration. Has two membranes. The inner membrane is highly folded to form cristae. The central part is called the matrix. Centrosomes and centrioles, organelles that are responsible for forming and anchoring the spindle fibres. Topic 3, membranes and transport. So if we look at the structure of the membrane, you've got an intrinsic protein that spans the whole phospholipid bilayer, has a pore. Extrinsic proteins are ones that lie on the surface. You've got the phospholipid bilayer, which is made up of phospholipids, which have a hydrophilic phosphate head and hydrophobic fatty acid tails that face away from water. You might have glycoproteins on the surface, which are carbohydrates attached to a protein, which are for cell recognition for like hormones. Glycolipids act as recognition sites, for example, for cholera toxins. And then you've got cholesterol, which adds strength and fluidity to the membrane. So membrane fats, they're partially permeable, made of phospholipids arranged in a bilayer and proteins. Under an electron microscope, you can see two dark bands. The fluid mosaic model describes the arrangement and movement of the molecules that make up a cell membrane. Fluid, phospholipids, and proteins are free to move sideways. Mosaic proteins have a scattered arrangement within the bilayer. Phospholipids, a molecule that is a constituent of the inner bilayer of biological membranes, having a polar hydrophilic head and a non-polar hydrophobic tail. Cholesterol, in cell membranes for fluidity and stability. Glycolipids are phospholipids with carbohydrates attached for cell signaling and recognition. Intrinsic proteins completely span the phospholipid bilayer. Some act as carriers to transport water-soluble molecules across the membrane, others are enzymes. Extrinsic proteins embedded in the bilayer on one side but don't extend through for cell signaling, cell recognition, enzymes and receptors. Roles of membranes within cells are for compartmentalization. They separate organelle content from cell cytoplasm, provide a large surface area for aerobic respiration, contain digestive enzymes. Where some reactions occur, the roles of membranes on the surface of cells separate cell components from external environment, transport of materials, contain enzymes for reactions, as antigens to recognize cells as their own, receptors for chemical signals. Cell signaling process. One cell releases a messenger molecule, for example, a hormone. This molecule travels to another target cell, for example, in the blood. The messenger molecule is detected by the cell because it binds to a receptor on the target cell's cell membrane. The cell then responds to the messenger molecule by a change in activity. Diffusion, the passive net movement of molecules from an area of high concentration to an area of low concentration down a concentration gradient across a partially permeable membrane through the phospholipid bilayer. Molecules that diffuse through the bilayer are lipid-soluble ones, small, non-polar molecules, for example, oxygen, very small, polar molecules like water, but very slowly. Factors affecting the rate of diffusion are temperature, concentration gradient, thickness of exchange surface, and the surface area. Facilitated diffusion now is a passive movement of substances from high to low concentration down a concentration gradient across the membrane, but it needs carrier or channel proteins. Channel proteins provide corridors that allow specific molecules or ions or charged particles across the membrane. Carrier proteins are proteins that transport substances across the cell membrane, like large substances like glucose. Factors affecting the rate of facilitated diffusion: concentration gradient, temperature, number of channel or carrier proteins. Osmosis is the movement of water from an area of high water potential to an area of low water potential down a water potential gradient across the phospholipid bilayer. Pure water has a water potential of 0 kilopascals. Adding solutes like sugar to water makes the water potential lower, less than 0, so negative. So the more negative, the lower the water potential. Water potential is measured in kilopascals. Active transport now, the active movement of ions or polar molecules from a low to a high concentration against the concentration gradient using a carrier protein. This movement against the concentration gradient requires energy from ATP. Facts about active transport. Molecules may move against the concentration gradient. Energy from ATP is required. Specific protein carriers are required. Change the shape to move substances from one side of the membrane to the other. Types of molecules that are actively transported by carrier proteins are large polar molecules or ions. What could inhibit active transport? Anything which inhibits respiration and therefore ATP production, for example, cyanide. Endocytosis, process by which a cell takes material into the cell by infolding the cell membrane. Exocytosis, the process by which a substance is released from the cell through a vesicle that transports the substance to a membrane to let the substance out. Phagocytosis, a type of endocytosis, and is how white blood cells, called phagocytes, engulf and digest bacteria. The end of that topic.